So, we are not going to have a big test in this class. We are not going to have a big project in this class. I wanted to spend some time going over some stuff that we talked about at the beginning of the year. And in order to do that without like actually giving you a cumulative final, I've decided to break it up day by day. So today we're going to talk about the scientific method because we touched on that very briefly at the very, very beginning of the year. Tomorrow is all about ecology, and I think I've got something fun for us planned there. Then we'll talk about cells, hit up photosynthesis, and then Friday we're just gonna put it all together. So, that is the general plan. There's a paper cut. Ooh, words. So, I actually want you to get out your notebooks. And just kind of take brief notes. You don't have to write everything down word for word, but just put your review content somewhere that you can find it pretty easily. Just so you're not just like sitting and zoning out. So at the very beginning of the year, I'm talking very, very beginning of the year, hybrid times, we were still using concept maps, very beginning of the year. We talked briefly about what biology was, and I touched on the scientific method. So we're going to go a little bit more in detail to review that process today. So can I tap this and have it move forward? I can. Okay. Oh my goodness, what did I do? Cancel. Goodness gracious. All right. We'll get there. Maybe. Yeah. Okay. So there are 6 main steps to the scientific method, right? 6 main steps. The first one is to question and observe. The entire point of being a scientist is to be curious and to ask questions about your surroundings. So before you can even start thinking of a hypothesis, before you can start designing an experiment, you have to be able to ask a question because the entire point of the scientific method is to find a systematic way to answer questions, to explore the world. So, the first step of the scientific method is to ask a question. And it's kind of hard to ask a question if you aren't paying attention. So, observe your surroundings, be curious, ask questions, pay attention. Once you have a question that you want to commit to answer, you have a question and you want to design an experiment or you want to carry out this process in order to find the answer, that is when you create a hypothesis. So, a hypothesis is a scientific explanation that can be tested. That's important. We can't make a hypothesis on something that can't be tested, either because we don't have the ability to, or it's just impossible. Like, we can't design an experiment about what it feels like for a human to walk on the surface of the sun, right? Because a human can't walk on the surface of the sun. Usually, these hypotheses are in the form of if-then statements. For example, let's see, if I were to give you a final this week, then there would be major mutiny, right? If-then statements. Something causes another action to happen. That gives us a hypothesis that we can easily design an experiment to test around. So once we have our hypothesis, once we have our goal statement, what we think is going to happen, what we are going to test, then we design an experiment, but not just any experiment, a controlled experiment. In a controlled experiment, we are only changing one variable. So 2 situations have to be exactly the same, but we only change one thing. If we were to change more than one thing, what would happen? I'm giving you a guess. Why don't we just change a bunch of factors? in an experiment? Why do we want to only change one? If we were to be testing what the best type of water for a plant is, right? Gatorade, salt water, or purified water, right? If we were to change the different type of water that we are watering them with, but we are also to put one in the sun and one in a cabinet, are we able to tell that it was the water that caused the plant to die or the lack of sun that caused the plant to die? Are we able to make that decision? No, exactly. So controlled experiments allow us to see or allow us to like actually know that what we are changing is causing an effect. So a controlled experiment has an independent variable and a dependent variable. The independent variable is what we are changing. So if it's the type of light we're giving a plant, if it's the type of food that we are giving a mouse, right? The independent variable is what we are changing. The dependent variable is what we're watching. The dependent variable responds to the changes we made in the independent variable. So if we're trying to see if a mouse likes one type of seed better than another type of seed, right? We're not watching the seed, we're watching the mouse. So the mouse's behavior is our dependent variable, and the independent variable is the seed that we are changing. So controlled experiments have independent variables and dependent variables. Independent variables we are changing. Dependent variables depend on what we're doing. Make sense? All right. So this also lends us to control groups. A control group has a change made to the independent variable that we are testing. So this is typically the standard, right? So if we were doing the what liquid is best to water a plant with, right? The control group would just be regular water. Or if we were to decide what type of light a plant likes best, right? The control group would be just like regular sunlight. This is the placebo group of medical trials, right? If we're trying to test whether or not a drug affects a human body in the way we want to affect it, and it's not all mental, so we have our regular group and we have our placebo group that thinks they're getting the medication, but they aren't. Right? That control group, the one where there's really no change, that's that placebo group. That's that control group. That makes sense? Okay. So after we have carried out our experiment, after we've done we've designed our groups, we've carried everything out, the next step is to record your results. You write down any changes, any measurements, any data. This is where the only difference between screwing around and actually doing science is writing it down. Because if you do an experiment with different types of water and different types of plants, right, and you don't write anything down, you don't collect your data, you're just gardening. But if you write it down, it can be science. So after you've got your results recorded, then you draw your conclusions. So you don't make any assumptions while you are collecting your data, while you are carrying out your experiment. You lean one way or another, but you can't really make a decision until you sit down and look at your data, and that's when you draw conclusions. That's when you use the data you recorded to summarize your results and either reject or fail to reject a hypothesis. That's a big thing with science, right? You can reject a hypothesis, be like, yes, my hypothesis was not true. Like if my hypothesis, if we're going back to the very, very beginning of the year, is that Gatorade makes plants grow, that if I water my plants with Gatorade, then they will grow to be big and strong. If I find my results actively kill those plants, I can reject that hypothesis. But scientists don't like the word approve very much. because a lot of factors can go into stuff and we can't really denote a lot of things as fact that easily. So we either reject or fail to reject a hypothesis. And finally, our last step is to communicate. What's the point in doing science if you can't tell the world about it? And there's a lot of different ways to do this. A lot of scientists write a paper. Some of them do poster presentations. A lot of them do both. Some of them have lectures or symposiums where they announce their scientific discoveries to the world. There are a lot of different ways to communicate data, to communicate your experience. So, I know it's a Monday morning, and I know that y'all zone out a lot, but can we do one of these together? Okay, like an inkling of participation. So I want to do one of these together. Oops, I skipped a step. Before we get too started, when you are presenting your data, graphs are a really good way to do that. It allows people to see what's going on rather than just like look at a big table of numbers. And when you are graphing the data that you collect, your independent variable always goes along the bottom, and your dependent variable always goes along the y-axis. So dependent variable is on the y, independent variable is on the bottom, on the x-axis. Okay, so we're going to do one of these together. You have 3 options. So I need at least one of you to make a decision. Which one are we going to do? Do we want to design an experiment about the type of fertilizers that helps plant grow? Do we want to design an experiment about our phones having more bacteria on them than a toilet seat? Or do we want to do an experiment about, or design an experiment about adding weight to a paper airplane, making it fly longer? Which one of those do you guys want to play around with? Yes. One? Anybody else? All right, fertilizers in plants. Okay, so what's our hypothesis going to be? If what, then what? What do you think is going to happen? So if we use fertilizer, right? What do we think is going to happen? I'm going to make a hypothesis. If we use fertilizer, what do you think is going to happen? Yeah. Plant growth. We think, okay, so if we use fertilizer, then our plants will shrink. That's our hypothesis. So now the next step after a hypothesis is to design an experiment. We need an independent variable. We need a dependent variable. We need our experimental group, and we need our control group. So, can I write on this? Aha, I can. Okay, so independent and dependent variables. So, independent variables are what we are changing. So, what's the thing that we are changing in this experiment? What's the thing we are affecting? The plants, what are we doing? Okay, so we've got plant growth and we've got the actual use of fertilizer. Which one are we changing? The growth, okay. What we are doing will affect the plant growth, which would make our plant growth our dependent variable. Which would make what our independent variable? That hurt. I think I heard it. A little bit louder. You got this. You had it. Plant growth is our independent, our dependent variable. What's our independent variable? What are we changing? Fertilizer. So we are going to try different amounts of fertilizer on a plant to see if it will grow or shrink, right? whether or not that plant grows, that's our dependent variable because it depends on what fertilizer we're using. So what would be our control group? What would be the standard to which we're comparing? Yeah. Okay, yeah, well, plant growth is what we're measuring. So if we're setting up an experiment, right, we've got a bunch of different plants. And we want to know if fertilizer will help it grow or shrink, right? So if we give all of our plants fertilizer, we have nothing to compare that to. Plants without fertilizer. So our control group is no fertilizer because we need that standard to compare it to. If we give all of our plants fertilizer, right, we don't have a standard. If they shrink, we wouldn't know because they would all do the same thing in theory. So our control group is no fertilizer. What's our experimental group? With fertilizer. Beautiful. So now we have our experiment, right? We know that we are going to have plants with fertilizer. We are going to have plants without fertilizer, and we're going to change nothing else. We're going to give them the same amount of water. We're going to talk to them the same amount. We're going to put them in the same spot so they get the same amount of sun. We're going to have the same size pots with the same amount of soil. But some of them are going to have fertilizer, and some of them are not. And we're going to watch plant growth based on our fertilizer. Cool? Following? Beautiful. All right. So now we have our experiment. We've carried it out, and now we have our data, right? So we have, let's say, the amount of fertilizer and our plant growth. So we have our y-axis and our x-axis. One of them had the independent variable and one of them had the dependent variable. Do you remember which one was which? Let's go back and find out. There it is. Our dependent variable will go on our y-axis, and our independent variable will go on our x-axis. Yes, dependent on the y, independent on the bottom. So if we were to if we were to actually graph our data, right? Say we had a chart, we had all of these numbers. That's our data that we've collected from our experiment. So our x-axis will be our independent variable. So this is going to be the amount of fertilizer, right? So this one's going to have 0 fertilizer. We put 1 gram of fertilizer, 2 grams of fertilizer, 3 grams of fertilizer, right? Different types of fertilizer to measure plant growth. And this is our dependent variable. So this would be our plant height. And this, we'll say, is our amount of fertilizer. So now we can see visually whether or not the fertilizer had an effect on plant height. Does that make sense? So we've got our data. How do we? Yes. So we've got our data in our graph. Do we want to write a paper? Do we want to make a poster? How would you, if you had this data, how would you want to communicate it? How do you think the best way to communicate it would be? give you a hint. There's no wrong answer. What do you think the best way to do this is? How would you tell people about this? Yes. Poster. Why would you use poster? Yeah, okay. So we got a poster. We can put our graph on there. We can put a big hypothesis on there. Give all the information you need in a short, sweet, to the point kind of thing. Doing okay? Okay. So now it's your turn. We have, I don't know, the clock's not on here, but what you are going to do for me today is choose a question, either a question that we did not use from this PowerPoint, or just another question that you want to design an experiment about. You're gonna ask a question, form a hypothesis, and design an experiment. All of this is laid out in a chart for you on Canvas. So if you open up Canvas, I will. Oh yeah, y'all have plenty of time. If you open up Canvas, there is a little chart for you to fill out. Nothing too crazy. Choose a question, write your hypothesis, do exactly what we did together. Cool beans? If you want the questions back up, I will do that as well. Does anybody have any questions for me? All right. If you think of a question, let me know. I'm more than happy to help.